In part 5 of the immune system, we're actually going to divide this up into two more parts, 5A and 5B. Part 5A is going to be over adaptive immunity, but this is going to cover the players of adaptive immunity, who the players are. Okay, so here we're dealing with adaptive immunity. So once again, we return to your flow chart, and specifically what we'll be focusing on now is cell-mediated immunity. Be sure that you remember that cell-mediated immunity involves T lymphocytes. Okay, so in a cell-mediated response, we're going to use different types of T cells. They're all T cells, but there are different types of them. These T cells are going to defend against invaders that hide inside of cells. So you may have virally infected cells. You can have cancer cells. You can even have intracellular bacteria. Now, with T cells, they have to come directly into contact with their target cell, the target cell being the virally infected cell or the cancer cell or the intracellular or the cell that's infected with an intracellular bacteria. And we'll describe shortly how those T cells come into direct contact with those cells. The types of T cells we'll be working with are the helper T cells, which are also known as, or AKA, CD4 cells, cytotoxic T cells, which are also known as CD8 cells, and then suppressor T cells. Be sure that if I mention a CD4 cell to you, you understand that that's a helper T cell. Okay, so let's look at the functions for these three different types of T cells. The helper cells, think about them as being the regulators, okay? They're the helpers. They regulate. They do not kill, okay? Remember that these are also called the CD4 cells. Helper cells will also secrete cytokines, and that's actually going to enhance the activity of your macrophages. Remember, the function of the macrophages is to phagocytose. Okay, so these cytokines will also enhance the activity of the natural killer cells. Remember that natural killer cells secrete perforins, which create a pore in the membrane of your infected cell. The helper T cells secrete cytokines, which will enhance the activity of B cells. And we talked about B cells in the previous part. Remember, B cells are important in humoral immunity. Helper T cells will also secrete cytokines that will enhance the activity of cytotoxic T cells, and we'll talk about those below. Then it also enhances the activity of other helper T cells, and it enhances the activity of a suppressor T cell. Remember what cytokines are. Back in Chapter 5, when you were introduced to communicators or chemical messengers or chemical ligands, cytokines are an example of a chemical messenger. Now, the cytotoxic T cells, remember these are also called CD8 cells, are directly responsible for cell-mediated immunity. These cells kill. They don't regulate like a helper T cell does. Cytotoxic T cell kills infected cells. It will kill cancer cells and it will kill transplanted cells. Then we have the suppressor T cells. And we won't spend a lot of time talking about suppressor T cells, but they do produce cytokines. So again, these are chemical messengers that are going to suppress the activity of the B cells. They're going to suppress the activity of the cytotoxic T cells. And they'll suppress the activity of helper T cells as well. So when you think about suppressor T cells, think about these in terms of turning off an immune response. Our main focus here is going to be on the helper T cells and the cytotoxic T cells. So we talked earlier about how T cells have specificity, but here's the catch, right? There's always more to it. Remember that the T cell has the T cell receptor, and we said that the T cell receptor has an antigen binding site. And this is all true. However, the only way that a T cell will bind to an antigen using its T cell receptor is if that T cell or the antigen is associated with an MHC molecule. So MHC molecules, these things are called the major histocompatibility complex. Okay, so these are called self-proteins. Everybody has MHC molecules, and everybody's MHC molecules differ from person to person. So these self-proteins are also sometimes called self-antigens. And these self-antigens consist of glycoproteins. proteins. All right, so they are a protein, but they've got a little bit of a sugar attached to them as well. So let's take a look at how these MHC molecules have to be associated with the antigen, which therefore have to be associated with the T cell. In this particular example, here we have at the top the plasma membrane of your cytotoxic T cell. This could also be a helper T cell. And on the bottom, we have the plasma membrane of an invaded cell. So maybe this is a cell that's been invaded by a virus, for example. What has to happen here in order for the T cell to bind to the antigen? So first off, we know that there is an antigen inside of this invaded cell. There's also an MHC molecule that's found inside of the cell as well. So what has to happen then is the MHC molecule has to bind to the antigen. Once it binds to the antigen, then this entire complex here gets presented on the surface of the cell. So here now what you notice is that you have the MHC cell, and then here's the foreign antigen. So this is the antigen that was inside of the cell, and now it's been brought to the surface of the cell. Once it's been brought to the surface of the cell, now the T cell will bind to the antigen via its T cell receptor. And then once that's done, something else will happen next that we'll talk about in a little bit. So this whole process right here is called antigen presentation. So what this means is that we have a cell. This cell is called the antigen presenting cell. So it's a cell that's presenting the antigen to the T cell. And now that that happens, the T cell can bind to the antigen. So let's take a little closer look at this MHC molecules. So there are actually two different classes, and I'm actually going to step those through with you. But what you are going to have to look up is the bottom bullet here. Are MHC molecules the same for every person? And what would this have to do with the skin graft or even an organ transplant? So let's go ahead and look at the different classes of MHC glycoproteins. We have two different classes. Class 1 is located in the plasma membrane of all nucleated cell. Class 2 are restricted to the surface of just a few types of immune cells. So these are macrophages, B cells, cytotoxic T cells, and dendritic cells. So let's kind of take a closer look at the relationship between these class 1 and class 2 glycoproteins and their T cells. So class 1, we said, is found on the surface of all nucleated cells. Class 1 are recognized by only cytotoxic T cells. So class 1 will only be recognized by a cytotoxic T cell. So cytotoxic T cells can destroy the body cells if invaded by a foreign antigen. So this is what will happen. Cytotoxic T cells will bind to an MHC class 1 molecule. Then its response is going to be to kill that nucleated cell. Then there's the class 2 MHC glycoproteins. These now are found on the surface of certain immune cells. Class 2s are only recognized by helper T cells. So a helper T cell will bind to an MHC2 glycoprotein. And when it does this, remember what a helper T cell does. A helper T cell regulates the immune response. So when the helper T cell binds to MHC2, it will regulate the immune response. Okay, so let's take a closer look at class 1 MHC and cytotoxic T cells, and then we'll come back to class 2. The cell on top is the virally infected cell. Remember that this could also be a tumor cell. It could be a cell that's been invaded by a bacteria as well. Okay, down below is the cytotoxic T cell. So because we're dealing with a cytotoxic T cell, right away you should realize that it will only recognize a class 1 MHC molecule. So let's look at the process of how this is going to work. We're going to start with a newly synthesized viral protein. So the virus has already invaded the cell and it's taken over the machinery of the cell and it's producing its own proteins. So then in step 2 here, the proteins become fragmented. So in other words, they start breaking apart. And then once they become fragmented, then in step 3 here, the proteins will bind to the MHC class 1 glycoprotein. So once that happens, then in step 4, the class 1 MHC molecule presents this antigen fragment, right? The antigen fragment being the viral protein, and it's presenting it to the cytotoxic T cell. So once this happens, then in step 5, the T cell receptor will bind to the antigen. So it's binding to the antigen that's bound to the class 1 MHC molecule. And then here we have in step 6, the CD8 molecule. So the reason cytotoxic T cells are also called CD8s is because the CD8 is a protein that actually ensures or enhances the binding of your T cell receptor to the antigen. Because once you've got it there, you don't want to lose that cell, right? You want to be able to kill that cell. You don't want to lose it. So in step 7 then, the cytotoxic T cell will kill the cell. It will kill that virally infected cell. So now that takes us to the class 2 MHCs and the helper T cells. So now what we're going to look at, down below is the helper T cell. At the top now, remember that class 2s are only found with certain cell types. So here, for example, we've only listed a macrophage or a dendritic cell. So in step 1, your phagocytic cell has engulfed the antigen. Remember, macrophages and dendritic cells are phagocytic. So they've engulfed the antigen. The antigen in this case are viral proteins. Then those viral proteins are fragmented. And then once they're fragmented, they're going to bind to a class 2 MHC molecule. And after they've done that, then we are going to present this antigen to the helper T cell. Then once that happens, then in step 5 here, the T cell receptor will bind to the antigen. Remember, the antigen now is bound to a class 2 MHC molecule. And then once that happens, the helper T cell is going to secrete cytokines to induce and regulate the immune response. Keep in mind here which cell is the antigen presenting cell. Okay, that's your macrophage or your dendritic cell. So next then we have helper T cell activation. And so these are things here that you're just going to have to commit to memory. So we already said that helper T cells are going to secrete cytokines, which activate the immune system. So they do not directly interact with the antigen though. Okay, so what helper T cells can secrete? They can secrete B cell growth factor, and that enhances antibody secretion. That's important, right? Because once you make antibodies, as you learned in the previous part, can do things like neutralization, agglutination, activate complement system, assist with natural killer cells, and so on. So that's important. These helper T cells can also secrete chemicals that attract neutrophils and macrophages. So remember, neutrophils and macrophages are very important in terms of being phagocytic cells. They can also enhance macrophage migration. So you need to have macrophages in the right area if they're going to be engulfing invaders. Helper T cells can also activate eosinophils and promote IgE development when you have a parasitic infection, like, for example, a roundworm or maybe a tapeworm infection. And helper T cells secrete IL-2. IL-2 does a number of things. IL-2 will stimulate the proliferation of helper T cells. So if you make more helper T cells, you can induce more of an immune response. They also help the proliferation of cytotoxic T cells. The more cytotoxic T cells you have, the more you can kill. And then the proliferation of B cells, because the more B cells you have, the more plasma cells you can have, which means the more antibodies and memory cells that you will have. IL-2, which is interleukin 2, also stimulates plasma cell development. Because remember, if you produce plasma cells, you can go on to produce antibodies. And then helper T cells, when they secrete interleukin 2, that also enhances the activity of natural killer cells. So realize that when we keep talking about these cytokines, interleukin 2, IL-2, is an example of a cytokine. So it's an example of a ligand or a chemical messenger that only ever travels through interstitial fluid. So this is where we're going to stop. And in part 5B, I'm going to start putting all of this together for you.